would like to give you an overview about polymeric drug delivery systems. Of course, we can talk about these for days, but of course, because of very short time for this lecture, I'll try to summarize and give you an overview about drug targeting. So I would like to highlight our interest in our laboratory in Polymer Research Group in Toronto University. So we are mainly interested in use of polymers for biomedical applications. Like for example, we are pioneer for antimicrobial polymers. We introduced it for the first time in Egypt, so alhamdulillah, we wrote a few review articles about this topic, and we have some patents also about this antimicrobial system. So polymeric drug delivery system is also one of our priorities. Drug targeting. We had a project before about control of bilharzia. We have also other interests like using some polymer waste in some other applications. One of our interests is the nanotechnology, which is the electrospinning of polymers. So transferring polymeric materials to nanostructured materials mostly for biomedical application. This is also for many applications, but we are focusing in biomedical application because we have done for the first time nanopolymers or nanofibers for drug delivery application, and we registered 2 patents in the United States about this topic. I'm not going to go for other things, but wound dressing is also one of our interests. So let's go very fast about what is the drug targeting. So if we say about drug targeting, that means we need to do send the target, send the drug to a certain place in the body. This is the most important thing, that you need to send your drug to a certain cell or a certain organ in the body. Of course, this has a lot of advantage because if you do this, you mean you're going to avoid the side effects, the other side effects for non-target organs. So drug delivery is, or drug targeting is the delivering a specific drug to a specific site in the body. So targeting of drug to a specific site, of course, provides several advantages. A lot of advantage over the non-target or the conventional drug delivery systems. There is a difference between drug targeting and drug delivery. Drug delivery, so that means you make a drug available for longer period of time. So if you take the drug, it cannot be absorbed in the body at once. It takes longer time. So this is very important field, of course, for many applications like diabetes, like heart diseases, or a lot of things. So you need to do drug delivery system. So you deliver drug over a longer period of time. But drug targeting, if you deliver a drug, a certain drug to a certain site. So if you want, for example, to deliver something to the kidney or to the liver, other organs are not affected. So there is so many advantages for this technique. So most important advantage is that you prevent the side effects of the you enhancing the drug uptake by target cells, also by enhancing the drug uptake by specific cells, not only permits the internalization of substance with low cellular permeability therapy, limiting side effects. So you limit the side effects. This is the most important part. Of course, you increase the efficiency of the drug. When you do drug targeting, so you increase the efficiency of the drug. And another important point, you avoid the side effects which can sure affect the other organs in the body. Here, this is a number of research publications over the last 5 years related to drug administration. So, of course, there is increased number of publications, especially in the field of the cancer. So this is just example from literature to see that there is, for example, 2,294 publications in the field of cancer. Because of cancer, of course, cancer problem is a huge problem, and a lot of scientists are keen about producing something for cancer treatment. Also, inflammation is important field. So these two major fields are in concern when you're designing a drug for drug therapy. So other advantages also allow maintenance of low blood to cell concentration ratio therapy, reducing therapy limiting side effects. Also use liver drugs via, for example, I will concentrate on drug targeting to colon because we have experience in this field. So delivery of drug via colon over numerous therapeutic advantage. Drugs which are destroyed by stomach, because when you take a drug, as you know, the colon at the end of the human canal. So there is only 2 ways. Of course, you take it oral, but when you take it oral for the colon treatment, and before it goes to the colon, it's already mostly destroyed in the stomach. The other way is you take, for example, Tabostril. So I think this is not accepted from the patient around the world. We had the projects from European community, and they were interested that can we do another way to target to the colon. I'll show some of our work in this field, but let's see why we do this work. So also, colonic delivery is also beneficial in the systemic absorption of the drugs, like nifedipine and other theophylline and other drugs. Also, it's a promising site. Colon is a promising site for systemic absorption of peptides, protein, because of less hydrolytic hostile environment in comparison with stomach and small intestine. Colon also is highly responsible site for absorption of poorly absorbable drugs. Also, treatment of colon diseases such as colitis and Crohn's diseases is more effective when directly delivered over the affected area directly. Also, it gives a unique feature of the intended site and protection of the active agent until it reaches the site. Because when you send the drug to the colon, so you avoid a lot of problems for the drug and for the non-target organs. Also, targeted drugs can overcome limitation of conventional applications such as unfavorable biodistribution, and this is the point I'm talking about before. And there is a various approaches that can use drug targeting, for example, cancer therapy and other diseases. So there is a lot of definition for the word targeting. So I would like to talk for 1 minute about the methods of targeting. There is 2 methods, one called active and the other called passive targeting. Active and passive targeting, for example, active targeting is defined as specific recognition of the molecular structure of the target. So you recognize the body recognize the molecular structure of the drug, and this, if this is the case, this is called active targeting. But the passive targeting describes recognition of a molecule by the system okay, or makes use of enhanced permeability and retention effect in tumor tissue, for example. For example, passive targeting based on the size. For example, if you want to target something to the kidney, you can, in this case, you can use nanotechnology. For example, depending on the size of your produced drug, so you can design the size to go through the blood system, circulatory system. Then, if you design the size, for example, for the liver, then it can circulate until it reaches the liver and then gets in the liver. But if it's bigger than the liver, it cannot get in the liver. So your targeting here is depending mainly on the size of your drug. So in this case, you need to design your drug according to the porosity of the tissue in the organ. So if for example, if you want to design something for the kidney or the liver, you need to know the right size for this drug to avoid the side effects, which is the main idea behind the drug targeting. So how to target to the colon? So the main idea here, because of the colon is located at the end of the human canal, so you need to protect. This is the main idea here. You need to protect the drug from the stomach and small intestine. So you need to design a drug and then take it orally, and then this drug should be protected from any media or attack in the stomach or small intestine. So this drug must be released in the colon from drug delivery system. So this is some examples about drug targeting to colon. You see this inflammation here in the colon. So the approaches that you can use the transit time to target to the colon. You can use the transit time. You can use special enzymes in the target organ or in the colon. We're talking about colon here. So you can use also targeting moieties. You have designed for your polymeric material or your drug. You can use targeting moieties that can target your drug to a certain cells. Here, there's some prodrugs and some drugs. So this is like a kind of drug strategy for colon targeting. Prodrugs are drugs, but in its current state they are not active. So it is a drug, but it is not active in its current state. Since you give it to the patient, and it started to degrade and start to release the drug. So it is a drug, but it is not efficient in its current shape. So it has to be taken and then it's hydrolyzed and release the effective material. pH sensitive system or pH dependent system, that one, like the one I'm going to show, is about, for example, we made a special polymer for coating of drugs, and these polymers were found are sensitive to the pH. When it goes to the acidic pH of the stomach, it's not affected at all. It's like a spoon going to the stomach, so it's not affected. So the drug keeps inside this coated drug. But it only works in the alkaline media of the colon. So start working. So this is one of the techniques I'm going to concentrate. These coats could be, for example, polysaccharides. Can do coat for the drug to target the colon by polysaccharides. Of course, polysaccharides are safe materials, but there is some polysaccharides are not affected by the acidic media of the stomach. So the drug can pass through human canal until it reaches the column without a peak. It's only started working on the column. So there is some known examples for these polysaccharide. Also, it could be affected by fermentable coating. This coating can be removed in the basic medium only, or could be like drug polymer or drug saccharide, polysaccharide conjugate. This is a chemically bonded drug with the polymer, or could be like an enteric coating. Chitosan is a known polymer, natural polymer. This is derived from shrimp and crabs. And this is a very safe polymer you can use for coating or for many other applications. There is other natural polymers you can use for coating of these material. For example, para-aminobenzoic acid and para-amino hippuric acid have been used as drug for the polymer. Here the main philosophy behind the coating. So you have your coating. The drug coated with a polymer is not affected in the stomach. So go to small intestine, is only in the colon. Started to break down the coat and then release the drug. Here, this is another system for time-dependent delivery to the column. There is some examples for the drug we have used as a drug, and we coated it with some polymers like this. This is part of our work. These polymers contain azo group. It's well known that these azo groups can be reduced in the column. We used the idea of the reduction of the azo group in the column, so we coated the drug with these azopolymers, and then when it goes to the column, it degrades, the polymer degrades, allowing the drug to be released in the site. This is another example from our work also, and this is the macromolecular broad drug. We call it broad drug because it's in its current state is not effective, but when this bond of azo bond break and releases the 5-amino salicylic acid, it starts as a drug, and this happens only on the colon. There's some results for the release from the drug coated, so it takes about 6 hours to release maybe about 20% of the drug content, so it can last longer time. This is also some other examples based on polysaccharides. And polysaccharides, as you know, is a natural polymer, and for example, you can use any kind of polysaccharide. You can use cellulose, and this is a very cheap source for coating. Here the idea what's happening in the colon. This polymer is like this as a group, and this is degrade in the alkaline media to release the drug, because the coat is fragile in the basic media. So here this is a summary for what we said. We concentrated about the pH dependence of the drug release, and this is a broad drugs, and this is coated. This is a coated tablets. This coated, you can coat any kind of drug to deliver to the colon, but the most important point is select the right polymer. And finally, I would like to thank my collaborators. I have collaborators all over the world, in Egypt, in UK, in Italy, in Japan, and United States. So without them, I cannot do this work. I cannot be here to present some of our data today. Thank you very much.